Good morning. It's a pleasure to follow all of these wonderful lectures and to share with you an update on our work using three-dimensional cultured equine bone marrow derived mesenchymal stem cells to treat inflammation in the joint. I think we've had an excellent introduction by our keynote address talking about the perils of osteoarthritis. My focus in this talk is really looking at how mesenchymal stem cells can be induced to be anti-inflammatory in order to treat early post-traumatic osteoarthritis before we end up having the pernicious effects of long-standing inflammation in the joint that we can see on the right here, which includes loss of cartilage, which hopefully very, very soon we'll be able to have this replaced in humans and in animals. But if we can intervene before it progresses to this point, perhaps we can actually avoid the more invasive and difficult process of replacing cartilage and have healthier joints for a longer period of time. It's very interesting that a lot of studies using mesenchymal stromal cells or mesenchymal stem cells to treat inflammation or even osteoarthritic joints hasn't been universally successful. And there's a lot of complication to the use and preparation of mesenchymal stem cells for treating joint disease or even inflammatory conditions. The reason for this is that when we're expanding and growing mesenchymal stem cells in monolayer, they're actually not at all physiologically induced. They are really more primed toward forming bone than they are to be anti-inflammatory. In terms of the mechanisms that mesenchymal stem cells use, it really is interesting to me, and I've thought for a long time that if immunologists studied MSCs before, molecular and cellular biologists, they'd be considered part of the immune system. And while I believe in tissue engineering and have taken that approach in some of my research, I think that the anti-inflammatory process that MSCs can be involved in is also quite powerful. A lot of the mediators to fight inflammation that MSCs use are sometimes thought to be pro-inflammatory, such as PGE2, not TSG6, but PGE2 and interleukin 6. Actually, it can have an anti-inflammatory effect on macrophages. And actually, in conjunction with MSCs, macrophages can be induced to make interleukin 10, which is a potent anti-inflammatory cytokine. Mesenchymal stem cells have also been shown to modulate T cells, dendritic cells, and neutrophils in their paracrine mechanisms of anti-inflammatory effects. As I mentioned, PGE2 is a rather surprising, although well-established anti-inflammatory mediator from mesenchymal stem cells, and TSG6 is another one. Interestingly, both interleukin-1 receptor antagonist protein and interleukin 10 have been shown to be inconsistently produced by MSCs, but have been produced. So, a lot of the work that's been out and summarized by Wang et al. In 2014 is that inflammation itself induces MSCs to be anti-inflammatory. And without that inflammation, you can actually very much reduce the anti-inflammatory effects of MSCs. So if you look at this sort of standard wound healing graph that was adapted from Wang, you can see that if you're following the healing process, at first when you create a wound or you have an injury, inflammation is peaked during the inflammation stage 1 phase and then tapers off gradually over time during the reparative and stage 3 remodeling phase. And if you were to graph the efficacies of MSCs in this environment, the efficacy is better during stage 1 than the later stage. So this is great if you're treating an inflammatory condition. However, if you're actually treating something like osteoarthritis, which doesn't have as much of an inflammation process, you can actually not have very good efficacy. I left out that you can also separately, without putting the stem cells into an environment that's inflammatory, you can use mediators that actually induce an anti-inflammatory state, such as interferon gamma, TNF-alpha, interleukin-1, and interleukin-17. How else might we be able to induce an anti-inflammatory state in our stem cells? So when you use these cytokines to induce an anti-inflammatory state, that's considered or termed cytokine licensing. And you also can induce an improved survival of the stem cells in models of systemic inflammation, and the animals themselves will survive better. So there's another approach, which is the approach that we took in our work, which is using three-dimensional culture of MSCs in order to induce an anti-inflammatory state. Others before us had shown that using three-dimensional culture for MSCs alters the expression of 710 genes and had the similar in vitro anti-inflammatory effects that you see in cytokine licensed cells, but without having to use those cytokines. In addition, there are a couple more recent studies in rats showing that the stem cells survived better in joints, stayed in the joints longer, and improved signs of osteoarthritis better than monolayer cultured MSCs. So our work that was recently published in Stem Cells and Development, just validating the formation of three-dimensionally grown MSCs from horses, showed that, first of all, if you are using a hanging drop technique to basically get your cells to aggregate, it takes about 72 hours for the cells to self-aggregate into these spheroids. It's just a sphere, but it's not a perfect circle, so we call it a spheroid. In addition, what I found to be very interesting, if we optimized to about 50,000 starting cells per spheroid, the viability was actually quite excellent. So even though the center of the spheroid you might think might have lower oxygen tension and access to nutrients, there was no coordinate decrease in the health of the cells. What was also interesting is that the spheroids did not continue to grow. I actually thought that when we started to do this, we would be able to use this in a spinner bioreactor similar to what you've already heard to basically expand the cells, but they halted in the cell cycle. And when we looked at certain MSC markers over time, spheroid versus monolayer, they were very stable. So, the difference here is, less than twofold, and no significant difference, except for GNL3, which is also called nucleostemin, and upregulation of GNL3 actually halts the cell cycle in G1S. So I thought that was quite interesting, and we are looking toward more of a molecular explanation of what's going on there. In any case, just looking at in vitro before the in vivo study, and I realize I'm already going to be late, we looked at what inflammatory and anti-inflammatory cytokines were influenced by spheroid growth versus a monolayer baseline. And this is just a summary because this work has been published, that we saw that spheroid culture alone, without other cytokine induction, increased PGE2, TSG6, IL6, SDF1, and although there was a lot of variation, IL10 was upregulated. If you add to this an inflammatory environment by using endotoxin or lipopolysaccharide, you will see a further induction of PGE2, IL6, and IL10. And then as an additional control, we looked at monolayer alone with LPS versus monolayer with no LPS. And we only saw an increase in PGE2 and TSG6. So how would we be able to use these cells? These little spheroids are about almost 300 microns. Would they survive going through a 20-gauge needle? Is this practical? So what we did is we compared viability after needle injection through 18, 20, and 22 gauge needles and showed that actually spheroids retained a higher viability and lower senescence. So those cells are very healthy in their little balls, very happy. So now I'll turn to our in vivo study, and we're still processing the data from this. So what we wanted to know was, would allogenic spheroid MSCs reduce the clinical signs and synovial fluid parameters of synovitis compared to control, which we used the same vehicle, or monolayer MSCs? So the equine bone marrow MSCs that we used were allogenic. We screened for an anti-inflammatory donor that was one of our high IL-10 producers, and our treatment was 20,000 stem cells per joint versus autologous serum as a vehicle. We randomized the treatment. 8 horses received spheroid, 7 horses received monolayer. And it was a bilateral model so that we can compare within the same animal, because so many of the joint fluid parameters are variable from patient to patient, and there, in my hands, a lot of variability in synovitis between horse to horse when you're using LPS as the inducer. So we used a bilateral model which has its own things, and we used 0.5 nanograms of LPS. One leg was treated with equine bone marrow MSCs, either spheroid or monolayer, and then the contralateral control had the autologous serum vehicle. Outcome variables included physical exam, systemic white cells, circumference of the joint. This was in the tibial tarsal joint, so hind limb. So circumference, comfort level on bending the tarsus, lameness, which was a major outcome variable for us, joint effusion, and then our synovial fluid analysis. I will skip going through the statistics because of time. So in the final analysis, what was really interesting to us was that the spheroid versus control, so left versus right, 8 hours post-injection, there was a significant reduction of synovial fluid total nucleated cell count. And in terms of spheroid versus monolayer, different horses against each other, 8 hours post-injection, there was a significant reduction in total nucleated cell count. In the spheroid group, all control limbs were lame. In the monolayer group, some control limbs were lame and some treated joints were lame. And when we looked at it as ordinal data, this was significant. We also looked at monolayer versus spheroid using our objective gait analysis data, which I'll show you. So the first bit of data I'll show are synovial fluid nucleated cell count, and at the 8-hour time point, we saw a significant reduction in total nucleated cell count, and this is standard deviation that you can see. For those who are not familiar with the lameness locator, perhaps this is a mistake, but I showed my raw data from 4 horses just as an example, that the spheroid-treated horses, the control limbs, the red and green shows lameness. You see quite a bit of lameness compared to the spheroid, which is in the same horse, which makes sense. And then the same thing in the other horse that you can see, it's significantly lame. And this is actually a very lame horse. So there's a lot of limping going on here. And then a couple choices from monolayer. There was one horse where the treated limb was significantly lame, and then there was another horse where there was mild lameness, so perhaps both sides are sore or neither side is all that sore. Now that's all well and good, but let's look at one of the horses. So this horse, who I actually adopted at the end of the study, I hope those of you who are not equine veterinarians can appreciate that yes, the right hind limb is quite lame in this horse, which was at the 8-hour time point, so not the significant one. And this horse was treated in the left hind limb and is perfectly sound because we ended up treating both with the stem cells at the end of the study. The push-off lameness, if we normalize the data, showed increased, and this is also standard deviation, not standard error, showed increased comfort in the spheroid, which is in orange, and then also in the impact lameness. So basically there's 2 different ways to measure soundness using this gait analysis system, and both showed improved comfort. We looked at joint circumference, but this was highly variable, and so there weren't significant differences over time. in terms of statistics. However, it's interesting that you can see basically if you compare the green to the purple, that's 3D control, so the spheroid control leg versus the spheroid leg, and it was always reduced in each of these, the averages, although there were a couple outliers, which I don't really understand. And then we had the opposite effect. So unfortunately, the statistics didn't hold true. Synovial fluid can be a little tricky for running cytokine analysis because of the large amount of high hyaluronic acid that's present. We're working on using Luminex for our data, but our early analysis shows no significant difference in IL-1. We have a whole bunch of other cytokines to analyze. In conclusion, activation of equine bone marrow mesenchymal stromal cells into an anti-inflammatory state seems to be very important for efficacy to treat inflammation in joints. And we think that it's superior to monolayer culture and hope to conduct some clinical trials. I have been using these both clinically in patients, and also I have conducted a study at The Ohio State University using this in dogs, so spheroid culture for dogs, and we're in the data analysis part of that. I'd like to acknowledge my funding, the Grayson Jockey Club, ACVS, and the American Quarter Horse Association. I forgot to say at the beginning that this work was conducted as part of Sophie Boger's PhD in my lab. She's the first author on the abstract and on the presentation, and this was her excellent work. And she's a colleague of mine at Virginia Tech now, and I'd be happy to take any questions.
Thank you so much. So first, thank you everyone for staying on time. That was perfect. Before we go outside for a little healthy snacks, one question, okay?
Sure, I love questions.
Thank you. My question is two-part, but quick. Are the spheroids that you grow uniform in size, and how many cells are in a spheroid?
Excellent question. So we, a lot of the work before this was left out of this presentation. So we actually did some pilot studies looking at, what is the optimal cell number, and it varies depending on what cell type you use and what species cell you use. If you use a hanging drop technique, we use 50,000 cells per spheroid, and they were quite uniform. So the variation was within 10% in size at day 72. But we have a high throughput technique where we don't have to do the hanging drop, which is extremely labor intensive. And those spheroids actually are quite variable. And so we used a filtration technique to get a range of cell types, or spheroid sizes. And so that's the approach that we take for our clinical cases. It's really fast and easy. It takes 3 days. Yeah.
Excellent talk. Thank you very much. How did you make them anti-inflammatory? That part I was missing. Maybe you said it and I missed it.
I'm happy to address that. So just by virtue of the cell-cell contacts in a three-dimensional environment induced an anti-inflammatory state. So no cytokines, no nothing.
But how do you know they were induced?
Oh, well, that was the published work, so I didn't get into all the individual data. I compared the output of the monolayer cells versus spheroid. And here, I can go back to that. Do I have time to just go over that slide again? Because obviously, not everybody got it. Ooh. This is my summary slide. So monolayer baseline, so it's not the graphs and everything. I don't like busy slides. Compared to monolayer, and this is in stem cells in development, so you can pull this, but in comparison to monolayer, PGE2, TGS6, IL-6, SDF1, and IL-10 were all upregulated. And then if you add LPS to the spheroids, you have increased PGE2, IL-6, and IL-10. And then monolayer with LPS compared to monolayer without LPS only increased PGE2 and TSG6. But yeah, hopefully, it's not very convincing because it's not raw data, but it is published. Yep.
Thank you so much. All right, so this is really nice to break. And please use this time outside with coffee and healthy food to mingle and talk some questions.